So today our focus is going to be on blood. We'll be talking about functions, different types of blood cells, how they're broken down, how they're recycled, as well as blood types. And you'll use this information to complete some of your blood type labs and other work associated with this. Okay, so we're going to start with kind of generics of functions of blood. So one of the main functions of blood is what we think about, which is transportation. Okay, in particular, blood will help transport gases like oxygen and CO2. Okay, so it'll help move oxygen attached to the red blood cells as well as CO2, and we'll talk more specifically about that in a little bit. In addition to transporting blood gases like oxygen and CO2, it'll help transport metabolic waste, things like urea, which is found in the urine, lactic acid, which is that buildup when you're exercising too much, and so you use up all your oxygen for ATP and you get that burn in your muscles because you're doing lactic acid fermentation. So it'll help move those metabolic wastes to the particular organs. That'll help eliminate them. Hormones, any kind of hormone. That's the definition of a hormone, is that it's released from the particular gland and travels to a target cell. So thyroid hormone, adrenocorticotropin hormone, any hormone. Enzymes, remember enzymes as a general rule are going to be proteins. Okay? And we find proteins in the blood, and again we help move them from cell to cell. Nutrients other than those blood gases, like glucose, would be transported through the bloodstream. The various types of blood cells, and we'll look individually at all the types of blood cells in a little bit, as well as the plasma proteins. And again, we're going to look in detail at those as well. But as it just kind of to give you some background, those will help regulate your blood volume. They help with blood clotting, fighting infections. Again, we'll look specifically at some plasma proteins in a little bit. Here, let me get you that stuff real quick. So the plasma proteins in general, they're going to help regulate blood volume. Okay, so help determine how much liquid is in the blood. Okay, they're going to be used for transport, like for instance the hemoglobin that's used to transport the oxygen. So they can be used to help transport certain substances. Plasma proteins are important for blood clotting, and plasma proteins also help fight infection, like the antibodies that we make. Okay, they help fight infections. And I'm going to run out of room to write the word infection. Okay, remember these are video notes. Okay, you can pause, you can rewind, you can go as slow as you need to make sure you get everything down. Okay. I also want to remind you, as we move further through these notes, every word you need, just like in class, every word that you need is not necessarily going to be on the slide. So it may not be to your benefit to just start frantically copying the slide and then start listening. Okay? It would probably be better for you to kind of write stuff down as we talk about it and pause the video if you need to. Okay, so some more functions of blood are regulation. It helps regulate body temperature, and we've talked about how it does that with the vasoconstriction, in particular in the skin, and the vasodilation of those blood cells there. It can help regulate pH, and that's what your picture is showing you down here. You don't need to memorize this. This picture is just here to kind of help some of y'all get a little bit understanding of that. Remember, pH is basically our acid levels in our blood, and acid levels are basically regulated through our blood with the gases, okay? With the bicarbonate and carbonic acid that is used to carry the CO2. Okay? And so basically you're going to have receptors in the blood that are going to measure pH levels. And when those pH levels leave this particular range there, you'll have organs start to kick in to help you with that. You'll start breathing differently to try to release more CO2, okay? And your kidneys will kick in. Again, you don't need to know how the pH balance works. This is just for those that are kind of questioning, like, pH, how can blood help with that? Okay, it also regulates our fluid electrolyte balance, right? Our fluid electrolyte balance mainly by maintaining the water balance within the blood as the kidneys filter the blood for urine. Our blood also will help protect us, in particular our white blood cells that most of us know help fight infection. We produce antibodies that help us fight infection, as well as clotting factors. Clotting factors are important to stop bleeding. Okay? And so we want to be able to stop bleeding when bleeding occurs so that we don't lose too much blood. So blood is made up of two basic components. If you'll remember, blood is a connective tissue, and all connective tissues contain ground substance and matrix. And so the ground substance for blood is our plasma, which is our liquid portion of the blood made mostly of water. And then the matrix portion of the blood is the formed elements, which are basically our solids. Okay? And we have three types of those. We have erythrocytes, which are those red blood cells, leukocytes, which are those white blood cells, and then platelets. And we're going to talk about each one of these specifically. We're going to talk about the erythrocytes, the different types of leukocytes, as well as the platelets individually. This is just to get you to realize that blood has two basic components: liquids, which is plasma, solids, which are the formed elements. These erythrocytes, leukocytes, and platelets. So hematopoiesis is the creation of these new blood cells. We do this in our red bone marrow, and essentially what we're going to make are stem cells. Okay? You can see, I know the picture isn't super duper clear, but that's what this says right here, hematopoietic stem cells. So from the red bone marrow, you get this hematopoietic stem cell, and that stem cell has the ability to differentiate into all the different types of blood cells. You can see it differentiating into the red blood cells, the erythrocyte, all these various white blood cells, the platelet cells here. Okay, these are also types of white blood cells over on the right. So hematopoiesis really is our vocabulary term that we need to make sure we get down, that it's the formation or the creation of these new blood cells. Okay, and it will make the stem cell that can then differentiate into all the others. So let's talk red blood cells specific. Remember again, you can pause, add, don't just frantically copy down these words, right? Stay with me and take notes about what I'm saying as well. The words are mainly there, so I made sure to have words that you may have trouble spelling out there for you. Okay, so red blood cells, their basic shape is that they're going to be tiny. You can see this picture right here focuses on their shape. They're tiny. They have no nucleus. They're what are called biconcave. You notice they have these indentions here in the middle. So they have no nucleus. Okay? And because what this does is this increases their surface area because the center collapses down like that, and it increases their flexibility so that they can fit through those narrow capillaries and stuff as they're trying to transport oxygen to all your various cells. So very small, no nucleus, okay? And their main job is to carry oxygen to the cells. And they're going to carry oxygen to the cells using the hemoglobin protein. The hemoglobin protein is where oxygen as well as CO2 both can bind to the red blood cell. The oxygen will bind to the heme portion of the red blood cell, which is the iron portion of the red blood cell, okay, in the lungs. So the oxygen will bind to the heme in the lungs, okay, so that it can then be carried to the tissue. The globin part, the protein part, okay, is where the CO2 will bind. Okay, and the red blood cell has a higher affinity for CO2 than it does for oxygen. So if the CO2 levels in our blood become too high, it makes it difficult for oxygen to be able to bind to the red blood cells. Okay, so the heme is where the oxygen will bind, which is the iron. The CO2 is the protein globin, the globin is the protein that the CO2 will bind to. Okay, erythropoiesis, so making sure we can spell this right here. Okay, so erythropoiesis is the regulation of our blood cell production, and it uses the kidneys to do that. Okay, so again, erythropoiesis, erythro being that red, okay, is the regulation of these blood cells using the kidneys. So what will happen is there'll be a decrease in the blood oxygen levels. And when the blood oxygen levels decrease, that's going to cause this hormone here called erythropoietin to be released from the kidneys. that erythropoietin is going to cause stem cell production to happen in the marrow. Okay, I'm going to erase some of this stuff, so if you haven't gotten the heme and the flexibility, surface area, globin stuff, make sure you get it because I'm going to erase it. Okay, so back to the erythropoiesis here. So I'm controlling my blood cell production using the kidneys. So I've got a decrease in blood oxygen, and so that causes the release of the erythropoietin. The erythropoietin, so when this gets released, this erythropoietin, that's going to trigger a stem cell production from the marrow, right? We saw that earlier. So that's going to increase that hematopoiesis, that stem cell production from the marrow, okay, which will then in turn increase my red blood cell count, which will then turn around and decrease my erythropoietin, turn it off. It's a negative feedback loop situation, right? So once I have my red blood cell count back up, the erythropoietin is turned off so that I don't produce too many of them. And my red blood cells last about 120 days, which if you did the quick math is about 4 months. Okay, so my lifespan of a typical red blood cell is about 120 days, and then they're broken down. We've got macrophages both in the liver, that's what this big cell is right here. Okay, we have macrophages, remember these are big cells that eat things, in the liver and in the spleen that will phagocytize or break down the red blood cells. That process is called hemolysis. Okay? Hemolysis is that rupture or that breakdown of the red blood cells. And so this happens on a regular basis so that we can recycle them. And what will happen is the globin protein is broken down into amino acids. And we're going to recycle those amino acids. Your body will use those amino acids to build up other proteins. And then that will also release the heme portion or the iron portion of it. And so the heme will be broken down into the iron components, which will go back to the iron components, which will be stored in the liver, sent back to the bone marrow to be used to make new red blood cells. And then the bilirubin component, there's your spelling right there, the bilirubin, which is used to make bile for the gallbladder. Okay? Bile is used to help break down fats. So the red blood cell will get recycled when its lifespan is done. Okay, so now we're going to shift over to white blood cells. So white blood cells have a unique ability to do what's called diapedesis. Again, spelled for you right there. You realize ped is foot, right? These white blood cells can actually leave the bloodstream. Your red blood cells cannot. They stay in the blood vessels at all times. But your white blood cells can exit the blood vessels so that they can get to bacteria, parasites, mold, things that are out at your actual cells. So that's a unique ability of the white blood cells that the red blood cells do not have. White blood cells are broken into two basic categories. We have granulocytes, which are the ones right here on the left, and then we have agranulocytes. If you remember, A means not or without. So granulocytes, when you look at them, you can see all the little dots. If you look at the neutrophils, the eosinophils, the basophils, see all those little dots inside of them? Okay, those are granules. So granulocytes have granules inside of them that you could see when you look at the microscope. Agranulocytes, remember, not or without, so they do not have any granules in them. So we have three types of granulocytes. We have neutrophils, which are active phagocytes. Okay, so they're actively going around looking for things to eat, to consume, to break down. Okay, these are our most abundant type of white blood cell. So we have the most neutrophils of any kind of white blood cell. Okay, our second type of granulocyte are what are called our eosinophils. Eosinophils are important in inflammatory responses. Okay, most commonly the allergic response. So important in inflammatory responses and parasitic responses. Parasites, worms, you're going to see an increase in eosinophils. So if you're sick and they draw blood and they look at a blood count, they're looking at these. Is there a spike in the eosinophils? Do they need to be looking at some kind of inflammatory reaction or parasitic reaction? Are our neutrophils spiked? They'll count these, and by looking at these numbers, it can help kind of guide them in what direction they need to go to see what's wrong. Okay, our third type of granulocyte is this basophil down here at the bottom. Okay, basophils produce histamine. Those of you that have allergies, you may take an antihistamine, right? So you don't make as much of this histamine, because histamine is released with inflammatory responses. Okay? And one of the things that histamine will do is histamine will cause more white blood cells to come to the area. So it'll summon more white blood cells. Okay? But it also triggers things like mucus production. Okay? So histamine is released from the basophils. Sorry about that if it just spun on you. Histamine is released from the basophils. Heparin is also released from the basophils. You see in your description down here it says contains histamine and heparin. Heparin is an anticoagulant. It's an anticoagulant, meaning it'll make it so the blood does not clot. And sometimes that is beneficial to you. Okay? An example of when it's used in the medical field is if you have an IV in, okay, where the needle is, your body will try to seal up that hole. And so a lot of times there'll be some heparin in there to prevent it from clotting around the needle there. Okay? And so you also see an increase in these basophils when you're having an allergic reaction or you have asthma flaring up, again because of that histamine release. Your A granulocytes, again, they don't have any granules in the cytoplasm. You'll notice how their cytoplasm looks clear, no little dots in them. And we have two basic types of these. We have lymphocytes, which behind our neutrophils are our most abundant. They're our second most abundant type of white blood cell. Okay, and we have two types of lymphocytes. We have B lymphocytes and T lymphocytes. B lymphocytes are going to make antibodies. We'll talk more about these when we do immune system stuff. So B lymphocytes will make antibodies. T lymphocytes can turn into natural killer cells, which can help fight cancer. And they also can make helper T cells, which can tell the B cells to make more antibodies. So again, you have two types of lymphocytes. B cells, which actually make antibodies. T cells, which can become helper T cells, which tell the B cells to make more antibodies. Or they can also turn into cytotoxic T cells or killer cells, which can help fight cancer. We also have monocytes, and that's going to be our last type of white blood cell. Again, it's an A granulocyte, and these are big, giant macrophages. You can see in that picture there, it's consuming things. Big, giant macrophages that do phagocytosis throughout your body. Okay, our last type of cell here is technically a cell fragment. Okay, these are our platelets or our thrombocytes. So these are really important with blood clotting. So these are small cell fragments, and again, their main job here is hemostasis. Stasis means to stop, right? So that is our fancy term for blood clotting or stopping the bleeding. I know, again, this picture is not super duper amazingly clear, but this actually is the same picture from your book. I pulled the same picture from your book, so if you wanted to pull up that or open your book, you could have that picture open while we were having this discussion here. So hemostasis, again, or blood clotting, has 4 basic steps. So the first is going to be that you're going to actually have the injury. So the vessel wall gets injured, and so that's going to trigger constriction. You'll notice it got a little bit more narrow here. Slow down the blood flow for a little bit. So some constriction and spasm is going to occur first. That constriction and spasm is caused by the release of endothelium. Okay, so endothelium, you can see it right here, step 2 in your picture. Endothelium causes that constriction and spasm, which is going to help reduce some blood flow. Okay. And because we have injury here, we have now exposed the collagen fibers. Those collagen fibers are normally, if you remember, your blood vessel walls, right? They're lined with endothelium, and then that collagen and elastin, that connective tissue, is underneath that endothelium. So if I've injured that blood vessel wall, then I've now exposed those collagen fibers that aren't normally exposed. That's actually a benefit because the platelets will stick to that. The platelets don't stick to the endothelium layer, but once that collagen is exposed, they can stick to that, which is what we want. That's step 2. So step 2 now, we have the platelet aggregation. The aggregation is just them basically coming and sticking together. So they will adhere to the sides, to the injuries, to the collagen fibers that are there. They're going to release chemicals. They're going to release other clotting factors, and those chemicals are going to attract more platelets. If you remember, blood clotting is a positive feedback mechanism, so it will continue and get stronger and stronger and stronger. And so as those platelets get stuck to that collagen, that signals the more platelets to come and to gather to the area. Okay? It also helps try to maintain that. It maintains some of that vessel constriction. So now we've got a lot of platelets gathered together in the area, basically blocking it up, which is important. And that's going to be our step 3. Now I've got this platelet plug formed. And so that platelet plug is going to help prevent any immediate, hopefully immediate loss of blood, slow that loss of blood there. Okay, and it's going to, now we're going to have the coagulation part happening. Okay, there's your spelling for coagulation right there. Okay, and so what will happen with coagulation is we've got these clotting factors, which are some of these proteins that are in the blood that have now been activated. And so the clotting factors combine together what's called thrombin and fibrinogen, which are present in your bloodstream all the time. We've talked about this before, how it's important sometimes to have things present, just inactive, so that they can very quickly be turned on, so that our response can be faster. So these clotting factors trigger the thrombin and fibrinogen to combine together, and they form what's called fibrin. Fibrin is kind of like your own stitches. Okay, so it makes this fibrin, which is like a thread almost. And the fibrin is going to help secure that clump of platelets down to the vessel wall, to the vessel injury. Okay, so it's going to kind of weave through that platelet plug. It's going to secure it and anchor it down, because we don't want that big plug to come loose and then be traveling through the bloodstream, because it could block something else later from receiving blood. That's what an embolism is. So we don't want that. So these clotting fibers turn on the thrombin, combine the thrombin and fibrinogen, which activates them into this fibrin, which acts like the thread. And so now we get step 4, which is going to be the actual blood clot formation. And so the blood clot formation here and the retraction, so it's going to get smaller. So we've got all kinds of blood cells that are getting trapped in the mesh here that was formed from the platelets and the fibrin. And then the clot will begin to shrink. And so as the clot begins to shrink, it's going to pull those fibrin threads closer together, which will bring the edges of the vessel injury closer together, right? If I've got this fibrin attached to the edges and I start shrinking, I'm going to bring those edges closer together. Okay? That's going to trigger the stop. That's gonna make our body stop releasing clotting factors, so that we don't get so big of a clot that it closes off the entire vessel. Okay? And eventually that fibrin will dissolve. Okay? Yes, this happens inside our body. This is also what happens when we cut and we form a scab. Right? That scab is the fibrin, platelets, red blood cells, white blood cells that are all trapped in there. Okay? And over time, right, that injury shrinks and shrinks and shrinks, because that fibrin is pulling the edges of the skin closer and closer together so that they can eventually completely close up. And when that happens, that fibrin gets dissolved. It goes away. Okay, so that's kind of our overview there of hemostasis. So what are we happening? It's a basic 4-step process. The initial constriction, spasm, exposure of the collagen fibers, the aggregation of the platelets and bringing more platelets over, forming the plug and the fibrin, and then eventually shrinking that fibrin down so that the whole thing seals up. So the last thing that we need to discuss is blood types. Okay, so we're going to look at the basic types of blood, the antigens, antibodies made. We'll look at positive and negative, which is Rh factor, on the next slide. You may want to pause it for a second and sketch out this table. That may help you take your notes a little bit faster. Okay, so our 4 blood types are type A, B. I don't even know why I wrote C on there. What the heck? Okay, A, B. Let me just cross that off. And then AB and O. Okay, so type A blood, okay, is going to have A antigens or surface markers on it. Okay, blood cells are going to have these antigens or these surface markers. Okay, and type A blood is going to have surface markers that mark it as type A, like for instance these little circle surface markers. Which means, okay, our body is not going to recognize, let's say A blood is shaped like circles and let's say B is shaped like squares. So our body, if I have type A blood, then I recognize circles, but I don't recognize squares. So my body makes antibodies. Okay? Antibodies are like your defenders here. Our body makes antibodies for type B because I don't know what those are. And so I want to be able to defend from those. Okay? Sorry again if it spun on you. Okay? And so that means that if anything with B surface markers is introduced into my body, I will try to attack it. So if I need to get a blood transfusion, I can receive type A blood because my B antibodies won't attack it, right? My antibodies for type A blood look like this, right? So they don't match with circles, but they definitely match with squares. So I can receive A blood and I can receive type O blood. We'll talk in a little bit why you can receive type O blood. What I will have is an agglutination reaction, which is a clumping. And that clumping is bad because it can clog stuff up and it makes it so blood can't get to organs. Okay, so I'll have a clumping reaction with B and AB. Again, because you can see these antibodies that are shaped like squares, right? B antibodies. I'm going to go with just oh, that's a mess. Okay, let me label this as anti-B, shaped like squares, right? This is a little A cell. This is a little B cell. Okay, and so if I have type B blood, I'm going to have B surface markers. I'm going to have those squares. And I'm going to make antibodies that are shaped like circles. So I won't attack myself, right? Because they don't blend to squares. So I can't attack myself. But I'm going to be able to attack anything that's shaped like a type A. Because I don't recognize that and I don't want it. So I can receive type B blood. I can receive type O blood. And I will have a clumping reaction with A and AB. If I have AB blood, that means I have both. If you think back to your freshman year genetics, right, blood is codominant, meaning I'm going to have both circle-shaped surface markers and I'm going to have square-shaped surface markers. So I'm going to have a mix of both. So I'm going to have both A antigens and B antigens. I don't have AB, and they're not like mixed together. They're not like half squares, half circles, although this one kind of looks like it. Okay. My antibodies then, I'm going to make no antibodies. Because if I made A antibodies, right, I'd attack myself and that would be bad. Same where if I made B antibodies, I'd attack myself and I don't want that. So I have no defenders, meaning I can take anything. I can get A, I can get B, I can get AB, I can get O, because I have no defense. Okay? I will have an agglutination reaction with nobody. Again, because I have no defense. Now, if I have type O blood, I have no surface markers now. I have no antigens. So hopefully you've caught on to there are only 2 types of antigens. There are A antigens and there are B antigens. And you either have one of them, none of them, or a combo of them. So if I have no surface markers, that I'm ready to defend against everything. I have A antibodies and I have B antibodies. I am not messing around, right? I am defending, defending, defending. So the only thing I can receive is type O, because type O has no antigens to kick my defenders into gear. Okay? Which means I'm going to have agglutination reactions. I'm going to attack B, A, and AB because I have defense prepared for that. So my universal donor here is going to be type O. Change the colors so the green doesn't blend in with the others. Okay, so sorry, the iPad and I were fighting about changing the color of the pen. So the universal donor here is going to be type O, because type O blood has no surface markers on it. So think of it, it's like sneaky, right? It's not going to upset any antibodies. There's no reason to get your defenders going because there's no antigens to make them say, oh, wait, hey, this guy's foreign, right? There's nothing that triggers that. My universal recipient is going to be my AB blood because it has no antibodies. Remember, AB blood has no defense. So anybody that wants to can go there because nobody's going to defend it. Right? And again, we'll talk about positive negatives on the next slide. So again, antigens are the surface markers on the cell itself. Antibodies are what your body makes to defend yourself from a foreign thing. So now let's add in RH factors. Okay, so our RH factor is the positive and negative. So if you are positive, like A positive, B positive, okay, that means you have RH antigens on your surface as well. So for instance, if I was A positive, I have A and I have RH antigens. I have both of those things on my red blood cells. Okay, so I make antibodies for B, right? I'm anti-B, and I don't make anything for RH, right? Because I don't want to attack my RH. But if I'm negative, right? So let's say that I had a B negative situation, or an AB, let's be AB negative. Let's make it a little bit more complicated. So I've got A and B surface markers, right? So I don't make any antibodies for those, but I don't have any RH. At first, I don't make any RH factor antibodies. But if I am ever exposed to RH factor, I don't know what to make this look like. I'm going to make RH antibodies. And where this really becomes an issue, it's an issue for transplants. So that's why they want to match positive to positive, negative to negative. It also becomes an issue for mothers. If a mother is RH negative and the father is RH positive, okay, so the mother is negative but the father is positive, that means the baby has a chance to be RH positive. And at first, it's not an issue, especially with the first child. It's not too much of an issue. However, during the process of childbirth, the mother and the baby's blood is exchanged. It's not exchanged when the baby is, just the nutrients are exchanged when the fetus is developing. But during the actual process of birth, blood exchange between mother and the baby is going to happen. So that means then if the baby was positive, the mom has now been exposed to that positive RH factor, meaning mom now makes these RH antibodies. So now if this couple goes to have a second child and that second child ends up being positive, mom could then attack that child. Okay, so don't freak out. Okay, they have a shot for this. If mothers are negative, they get a shot. It's called a ROGAM shot. Okay, that suppresses that response, and so they don't attack that. Okay, but that's something that they do take into consideration is blood type of mother and father, so they can pay attention to those RH factors, that positive and negative. So let's look at the examples that we have over here on the side and see if we can kind of run through this. This is something we would do in class. where you would have them, where we would do this clumping. You guys would do the clumping and you would decide what blood types they were. We do have a virtual lab ready to go, hopefully, where you guys will be able to do this virtually. But I want to kind of look at it right now. I know the edges of the picture are a little bit blurry, especially on top. These are the headings of what's on top right there: anti-A, anti-B, anti-RH. So let's look at patient A. So basically what happens here is you put a few drops of the patient's blood in all of these different wells or samples. That's what each little circle, is a well where we put a little bit of the patient's blood. And then we're going to drop in anti-A, anti-B, and anti-RH. So, and we see either a clumping or an agglutination reaction, or we see nothing happen at all. Like, let's start here with patient B. Let's start on this one. Okay, so here's a sample of the patient's blood in this first well. And we put an anti-A serum, which means I put in A antibodies. And if I look at this reaction right here, nothing happened. So I put in A attack guys and nothing happened. That means that this person's red blood cells doesn't have A on it because we would have attacked them. So then I took a different sample of the patient's blood and I put it in another well, and then I added anti-B. Okay, so now I added these B antibodies. And again, I have no clumping reaction, meaning I don't have any. There are no B antibodies on their surface. So that tells me that this patient has type O blood. So they have type O blood because they have no surface markers, right? And then if I look in the final one, I put anti-RH, right? I put RH antibodies. Again, I had no clumping reaction. That means they don't have Rh antibodies. That means this person is O negative. Okay, so in an ideal world, you'll pause it and you'll try to do the other three, and you'll turn it back on and you'll see if you got them correct. Okay, so hopefully you paused it and now you're checking to see if you got them correct. So let's look at patient A. So in patient A, I have a definite clumping happening here in the well, which means I had A surface markers and I attacked them. I'm also getting a clumping here in the B well, meaning I had those and I attacked them. And I'm getting a clumping in the RH, meaning I had that. So this patient would be AB positive. Patient C, right? I put in my A antibodies, I got a clumping reaction. I put in B antibodies, no clumping reaction. And I put in RH factor, no clumping reaction. So this patient would be A negative. And my final patient, patient D, I put in my A antibodies, and I did not get a clumping reaction, so I don't have those. I put in my B antibodies, and I did, so I have those. And I put in my RH factors, and again, I had a clumping reaction. So this patient would be B positive. And again, hopefully we'll get a virtual lab going with this for you next week. These are all of your notes that you need for blood.